In addition to magnification, resolution, and refraction, contrast is also a challenge for microscopy. The lack of contrast is because we have a glass slide and then organisms on that glass slide, which are often transparent. So as we can see here, we're seeing very little detail associated with these microbes. We can change that a little bit by lowering the light and some other types of settings on our microscopes, for example, phase contrast, dark field, etc. But still, contrast is a challenge. However, if we can stain our specimen, as we have done here, we suddenly see a whole range of detail appear. These dyes then help increase the contrast for us to help us visualize our specimens. The dyes have affinity for cellular materials, and these cellular materials either have a positive charge or a negative charge. The ones used here have a positive charge, and those are by far the most common types of stains that we'll see in our lab. Positively charged stains are known as basic dyes, and they combine with negatively charged cellular components. So they will be attracted to anything that's negatively charged on the cell surface. Examples that you'll see in lab: crystal violet, saffronin, malachite green. We also have acidic dyes. And these are negatively charged dyes that are attracted to positively charged cell constituents, such as India ink. So India ink is a great example of an acidic dye. Using one stain, as we are demonstrating right here, this is just crystal violet. This is referred to as a simple stain. And it allows us to determine presence, i.e. if something's here, and also the shape of our microorganism. We can add more stains, multiple dyes together, and do what's called a differential stain to get more detail. We've got 3 examples here, so let's take a first look at the first one. This is a gram stain, and in this gram stain, we have purple and we have some pink cells in here. This allows us to differentiate between gram-positive and gram-negative specimens. These have different cell envelopes. The gram-positive are purple, gram-negative are these pinkish red. Then we have an acid-fast stain, and what this is looking for is a specialized compound in the cell wall of the organism called mycolic acid. And this is used for mycobacterium. So Mycobacterium tuberculosis, that causes tuberculosis, Mycobacterium leprae, that causes leprosy. Pink ones here are the Mycobacterium. The blue ones are the negative organisms or the negative cells. The third one here is an endospore stain, and we'll learn more about endospores later this semester. Endospores are survival structures for bacteria. The pink rods are negative structures, and the green circles or green ovals here are the actual endospores. So for example, Clostridium tetani, we would be able to look and see if somebody has tetanus and identify it using this particular stain. The gram stain is the most common microbiological stain. The first step in this process is to heat fix our specimens. We do that by passing the slide through a flame, and that sticks the organisms to the slide so they'll remain through our entire methodology. Once we do that, we then add crystal violet, which is a basic stain, and crystal violet will stain everything. Doesn't matter if they're gram-positive or gram-negative, everything will now be purple. The next step in this process is to add iodine. Iodine is what we call a mordant. A mordant stick things to other things. They're used in dyeing a lot. This aggregates the crystal violet molecules. So that means they stick them all together. So we have lots and lots stuck together instead of being separate from one another. And that then creates an insoluble complex that will bind better to the cell. All the cells still purple at this point. The next step is to decolorize. We do that with alcohol, and that will dissolve all the lipids found in the cell envelope. So it will dissolve all the lipids. And as it dissolves the lipids, it also removes any dye that's attached to those lipids. Now, that means that our gram-positive cells do not have an outer envelope that is made of lipids, so they stay purple. But our gram-negative cells that do have this outer envelope that is lipids are now going to be colorless. Now, again, we have this problem, colorless against a colorless slide means it's really hard for contrast. So the next thing that we do to improve our contrast is we add a color. So we counterstain, that's the scientific verbiage here. We counterstain with safranin, and safranin is this pinkish red dye. Again, it's another basic dye, and it will stain all of the cells pink that were colorless. So every cell now will be either pink or purple. If they're pink, they're gram negative. If they are purple, they will be gram positive. So pink, gram negative, purple, gram positive.